So welcome everyone to the 6th webinar in our new Biochemistry Focus Webinar Series, brought to you by the Biochemical Society and Portland Press. Topics in the series will cover different areas in the molecular biosciences and practical sessions to support career development. Suggestions for future topics and speakers, please see the website for more details. Those suggestions are welcome. So I'm Augustin Amour. I'm a project leader at GlaxoSmithKline. I'm based in Stevenage, and I'm here with my functions in the Biochemical Society. So I'm a member of the Clinical and Translational Theme Panel, as well as a local ambassador representing the Biochemical Society at GlaxoSmithKline. And please see more details about how you could be more involved in the Biochemical Society on their website. So I'm honoured today to present to you Professor Dame Kay Davies from the Muscular Dystrophy UK Oxford Neuromuscular Centre. The title of her talk is From Diagnosis to Therapy in Duchenne Muscular Dystrophy. She is the recipient of the Biochemical Society's 2020 Centenary Award. This award recognises biochemists of distinction from any part of the world and is awarded annually. It was established in 1961 as the Jubilee Lecture to commemorate the Society's 50th anniversary. This changed then to the Centenary Award in 2011 to celebrate the Society's 100th anniversary. Kay was awarded a DPhil in the Department of Biochemistry at the University of Oxford. After that, she had a Royal Society's Fellowship in France and joined the St. Mary's Hospital Medical School to study Duchenne Muscular Dystrophy, or DMD, a progressive muscle wasting disease. In 1984, as an MRC Senior Fellow, she moved back to Oxford to work on the molecular analysis of neurological disorders. She was appointed Professor of Genetics and then Dr. Lee's Professor of Anatomy, and set up the MRC Functional Genomics Unit to apply genomics to genetic disease analysis. Her own group focuses on the development of therapeutic strategies for DMD. Kay's earlier work paved the way for the use of mini-dystrophin gene delivery using adeno-associated viruses and contributed to the exon skipping approach to therapy. She discovered the dystrophin-related protein Utrophin and demonstrated that increased levels of Utrophin can compensate for the lack of dystrophin in a DMD mouse model, or MDX. This has led to the identification of small molecules which increase Utrophin expression in adult MDX muscles and alleviate the pathology. Kay co-founded the biotech Summit Therapeutics to translate some of her work into the clinic in 2003. She is a founding fellow of the UK Academy of Medical Sciences and was elected a Fellow of the Royal Society also in 2003. She was a Governor of the Wellcome Trust from 2008 to 2017 and its Deputy Chairman from 2013 to 2017. Kay was made Dame Commander of the British Empire for Services to Science in 2008. Kay's Centenary Award lecture today will provide a summary of the current status of DMD therapy with a particular focus on those genetic strategies taken forward to the clinic. Please send us questions during the talk for Kay by typing them in the question box as shown in the image on the screen. I will try my best to read those to Kay at the end of the presentation. And now it's my honor to pass on to Kay for the lecture.
Thank you very much, Augustin, for that introduction. And it's a great honor for me to present this webinar today. And I'd like to do it on behalf of my group and all the workers that have worked with me over the last almost 30 years working on DMD therapy. I'd also like to thank the nominators and the awards committee at the Biochemical Society. As Augustin said, what I'd like to do is to tell you what the exciting developments are in the diagnosis and therapy of muscular dystrophy. In particular, we're now so incredibly close to getting approved really efficient therapy for muscular dystrophy that this is really a very timely lecture on this subject. For those of you who are not familiar with the disease, it's an X-linked progressive muscle wasting disease which affects only boys. Occasionally females are affected, but it's relatively rare. It's a tragic disease because it leads to early confinement to a wheelchair at about the age of 12. There's both skeletal and cardiac muscle involvement, and many of these boys die of cardiac involvement as well as respiratory failure. Some of the boys have cognitive difficulties, but I'm not going to address that particular issue today. Its prevalence is about 1 in 5,000 births, which means in all populations around the world, there are about 250,000 boys that are affected. At the moment, there's no curative treatment, only supportive approaches. But you can tell the difference that diagnosis has made in the field, because when I first started 30 years ago, boys generally died in their late teens. Now, some of them survive into their 30s or 40s, but in general, they die in their 20s. I became interested in this disease because it has a very high new mutation rate in all populations of the world, which means even if you could screen every pregnancy, you would not be able to eradicate the disorder. It's relentlessly progressive, and this photograph is taken of 3 brothers published in Victor Dubovitz's book. Diagnosis may be late in families that have never seen the disease before, which is where the new mutation rate is relevant. As you can see, the 2-year-old brother shows no outward symptoms of the disease. At 10 years, you can see the beginning of scoliosis, and you can see the extreme scoliosis 2 years later in his elder brother. So the tragedy is these boys are born in these families long before they are aware that they're going to be affected by this genetic mutation. It's the largest gene in the human genome. It encodes a 427 kilodalton muscle protein, not the largest protein in muscle, but incredibly, it is the largest gene. It covers more than 2.5 megabases of DNA, which in part explains why it has such a high mutation rate. The mutation causes a complete absence of the gene dystrophin, christened by Luc Hunkel, who discovered the gene dystrophin because the lack of it causes dystrophy. And you can see here in this slide at the bottom, normal muscle biopsy with dystrophin at the muscle membrane, uniform staining around all the muscle fibers, and in DMD, complete absence. Now, there is a milder form of the disease, first described by Becker, called Becker muscular dystrophy, where you get a truncated, smaller, semi-functional dystrophin. I'll come back to this in the context of mini dystrophin genes. And what you see in this particular biopsy is that the partially functional dystrophin can bind the membrane, but clearly not as efficiently as in normal individuals. So with a lot of work, biochemistry work from mainly Kevin Campbell's group and Azawa's group, we now know that dystrophin provides a vital link between the extracellular matrix and the intracellular actin cytoskeleton. Dystrophin itself does not lie across the membrane. It binds to this dystrophin-associated complex you can see here. If you take dystrophin away, then you lose that complex and you gradually get the muscle degeneration that you observe in patients. So it's now well established that dystrophin does provide this very important missing link. It's got this N-terminal region that binds F-actin, the carboxy terminal that links it across the membrane, and this rod structure in the middle made up of spectrin-like repeats. So that makes it a really challenging disease to develop therapy. It's a large protein, it's a structural protein. There's not a single genetic mutations. There are many that are known. The mutations are heterogeneous, but mainly deletions. You need to replace about 40% of normal levels for complete rescue. That's in contrast, for example, with cystic fibrosis, where you only have to replace a few percent of the wild-type ion channel to get a clinical effect. You need to target all muscles, skeletal muscle, diaphragm, skeletal muscle, and the heart. As I mentioned, cognitive deficit in 30% of patients. So those therapies that might target the brain as well are going to help. Muscle can make up 40% of a person's body mass, and therefore the challenge to get all of that muscle with replacement dystrophin is really been an issue in developing therapies. And of course, for any therapy, you need to avoid the immune response, and that's particularly relevant when you come to gene therapy. Now, I'm only going to discuss, as Augustin said today, genetic approaches. There are many, and this, you don't have to read this slide. This is just to remind me to tell you that there are lots of therapeutics that address the secondary effects of this disease: oxidative stress, inflammation, necrosis, fibrosis, and so on, and used in combination with genetic approaches in the future. I'm sure these will make a substantial difference. What I would like to concentrate on is not only just genetic approaches, but genetic approaches that are applicable to all of the patients. DMD patients, 66% of them are actually deletions in the gene. And those deletions occur predominantly in two hotspots, one near the 5' end of the gene and one near the 3' end of the gene. And that has enabled exon skipping approaches to be developed, because one particular set of oligonucleotides can be applicable to up to 13% of patients in one case, even though their mutations are different, so-called exon 51 skipping. And these sort of approaches have been pioneered by companies like Sarepta, and they are now approved in the clinic, but they don't work very efficiently, and at the moment they don't target the heart. But this is very much in development, and new exon skipping strategies are being developed. There's also stop codon read-through pioneered by PT Therapeutics, where again, they can achieve some efficacy in this, but both of these approaches thus far have only resulted in slowing down the disease rather than being a real effective treatment. I'm now going to focus on gene therapy and the modulation of utrophin, as these are two approaches which will not only replace the dystrophin gene with something that's partially functional, but also can be applicable for all patients, whatever their mutation. It's probably also important to note that both of these therapies will be applicable to both BMD, the milder form, where they produce some dystrophin, as well as the DMD patient, where no dystrophin is formed at all. The problem, as many of you will know, with using a virus that is commonly used to infect muscle, AAV, is that the dystrophin gene is very large. In fact, it's too large for the capacity of an AAV vector. So this led to the development of dystrophin mini genes, where we discovered, well, I won't say we didn't discover the patient. A patient was sample was sent to us by the late Sarah Bundy in Birmingham, and she expected this particular patient to have a very small mutation in his gene. This is him before he's just about to go off for a walk, and he's just left the army. You can see him against the army truck. This is him after he's just climbed the cliff, but you can see he needed a walking stick. This was in the early 40s. This is a few years later. This is him a bit further on in his 50s, where you can see he still needs that walking stick, and he's beginning to lean slightly back. So you can see from his gait that he is noticing the difference in his effective muscle, particularly in his legs. Now, this particular patient lived till he was 73. I went to his 65th birthday party, and it turns out that he has a remarkable deletion. We were amazed to find that he has 46% of his dystrophin missing. So the first question we asked, if he's got that much of his gene missing, perhaps he can naturally have another gene in his genome, which is compensating for the lack of his dystrophin gene. But you can see from this pedigree, this is the proband here, that he does have relatives with similar presentations. This one was dead, and this individual here volunteered a muscle biopsy. He was a weightlifter. He'd only been to his neurologist with mild muscle symptoms, and we were able to analyze his muscle biopsy. Now, this is the control muscle biopsy at the top, and you can see the abnormal muscle biopsy in the places where it's positive, and you can see he has the N-terminal and the carboxy-terminal end of the gene present, that is completely absent for this spectrin-like repeat, and therefore what he's actually doing is producing a mini version of dystrophin that clearly does bind to the membrane, not as effectively as wild-type dystrophin, but nevertheless effective enough for him not to have turned up in his neurologist clinic until his 40s. So this opened the way to treat muscular dystrophy, and many labs now around the world have nibbled at his mini gene to produce microgenes which will fit into the AAV vector together with muscle-specific promoters. So here's a schematic of the full-length dystrophin. This is the BMD patient I've just shown you. And various labs, Chamberlain's, Duan, Zhou's lab, have pioneered the development of these microdystrophins. And some of these are now incorporated into AAV vectors and are in clinical trials. They all contain parts, very important parts of the of the spectrin-like repeat region, known as the hinge region, but not all of them have the same hinge regions. You compare the Sarepta version to the Solid Biosciences version. NNOS is thought to be quite important for the function of muscle, but again, the very mildly affected patient didn't have this domain that binds NNOS, but nevertheless, in this particular microgene, the Solid Biosciences group were able to add this back in and engineer it in the hope of getting a fully functional mini gene. Pfizer have also presented a microgene, and the structure of that is shown here. The general message is that we need to have a reduced microgene in order to get function. We're not absolutely sure which of these domains is the most important. We know that certain hinge regions are critical, but whether NNOS, whether this particular hinge region is critical, remains to be seen. So what are the status of the clinical trials that I've just mentioned? Pfizer has started a clinical trial at very high doses, 10 to the 14 viral units per kilogram, and they have not published anything yet, but recent reports show that they are getting positive fibers in treated boys, and they are showing some effect, but that some adverse effects are observed, although these patients recovered within 2 weeks. This trial is continuing. At Sarepta, they've had quite dramatic results from their phase 1 trial, which has now been written up, and they see a dramatic clinical effect. Some of the boys show amazing ability to be able to get up from the floor easily and even to walk quite quickly in ways that they wouldn't have done without this treatment. Just 2 days ago, the FDA designated Sarepta gene transfer for a fast-track review, and they're currently undertaking a double-blind, placebo-controlled study of this particular mini gene. Hopefully, we'll get those results in 2021, but these look particularly promising in the context of being able to treat all patients, except for those that already have antibodies against the AAV. Solid Biosciences have done a dose escalation, and in their lower dose, they only saw low levels of dystrophin, which demonstrates you do need these very high titers of virus. They're currently on a clinical hold because they had some adverse effect in the high dose trial, but hopefully their trial will be continued again soon. So what are the challenges of AAV gene therapy for DMD? The first is the immune response. Some patients, 17% of them in fact, had antibodies against the virus that the Sarepta trialists use, for example. And they are trying to develop ways around that to reduce that immune response so that this particular virus can be used in all patients. Will we be able to administer this virus, but because perhaps after 5 or 10 years, the virus will be lost as muscle regenerates? There are challenges to prepare high titers of virus in sufficient quantity at the moment. So whether this is applicable to all patients is very good, but whether we can actually get this to all patients everywhere in the world remains to be seen. These sustainability of effect will be important. These viruses are administered in this time window, therapeutic window between 4 and 7 years of age. If you go in too early, will you lose the virus because of the growth of the muscle? If you go in too late, there may not be enough muscle to rescue. So we've been thinking of other ways besides gene therapy to compensate for the lack of dystrophin. And in 1989, Don Love, who was then working in my group, while we were trying to clone the dystrophin gene, identified a gene that didn't map to the human X chromosome, but mapped to human chromosome 6. It was ubiquitously expressed, unlike dystrophin, which is only expressed in muscle and brain. And you can see here from this schematic that these two proteins are very simple. And in fact, we know that they are related by an ancient genomic duplication event. But strikingly, the homology between these two proteins is greater than 95% in the actin binding domain and in this carboxy terminal domain, which binds that complex of proteins at the membrane. There's less conservation in the rod domain, but perhaps there's less evolutionary pressure here, as we know that there are many, not just the patient I've described to you, but other patients that are deleted across this region who have relatively mild disease. So is there any precedent? Do we see cases where utrophin can replace dystrophin? Well, if you look, this is taken from Tomey et al. In 1994. If you look at the developing human fetus, you can see very early on in human development that both dystrophin and utrophin are present at the membrane. In fact, we're not quite sure which comes first. As you progress towards birth, dystrophin is upregulated, and you can see it uniformly binding the muscle membrane here. And gradually, as you go towards birth, utrophin is downregulated, and in fact is only found in the neuromuscular junction in adult muscle. And this just is in DMD muscle, there's the normal, there's utrophin at the neuromuscular junction. And in DMD muscle, that's just one of the exons, naturally exon skip fibers, which is positive for dystrophin, which are relatively rare in DMD patients. But you do get, in the absence of dystrophin, you get a natural spreading of utrophin, not enough to show real clinical effect, but you can see that the utrophin starts to not just be localized at the NMJ, but to begin to occupy those absent sites at the sarcolemma. A study in 2006 by Cleopa et al. Showed that the amount of natural occurring utrophin appears to correlate with the disease severity, suggesting again that this may be a replacement. So our hypothesis is that in a normal individual, you have a developing fiber, and when you have mature healthy fiber, you've got dystrophin. When you've got a DMD fiber, in the absence of dystrophin, if you get some utrophin, if we could add something that modulated the expression or recapitulated the fetal program, then we might be able to compensate for the lack of dystrophin. So our first question was how utrophin compensates for the absence of dystrophin. Does that help in the MDX model of the disease? How much utrophin do we need? And if we increase utrophin with a drug, which is going to be everywhere, will we have side effects? Well, the first experiment in the MDX shows quite dramatic effects, that it indeed, overexpression of utrophin prevents the disease in the MDX mouse. And in fact, it also does that in the in the dog model of the disease. Recent data from Hansel Stenson's lab using a utrophin rather than a DMD mini gene and viral delivery demonstrates very efficacious expression of utrophin and dramatic rescue of the severe muscular dystrophy in the dog. We have also shown that we don't need to increase utrophin very much, and I'll come back to this at the end of my talk. Less than twofold increase provides benefit in the MDX mouse, and if we overexpress in all tissues using a ubiquitous promoter in the mouse at levels that will rescue the disease or prevent the disease, then we don't have any deleterious effects. And that's been shown for dystrophin also by Jeff Chamberlain. If you overexpress dystrophin, you don't get deleterious effects either. So this just shows you the localization of utrophin at the muscle membrane in two transgenics, Fiona and Freddy, compared to the MDX. And I'm putting this up just so that you know, if we look at the percentage of centrally nucleated fiber, which is the indicative of the disease in the EDL and the diaphragm, you can see that very high levels of expression in a transgene like Fiona rescues or prevents the phenotype in the MDX mouse, whereas Freddy, much lower levels, but uniformly distributed across the fibers, also gives substantial benefit in preventing the disease. And you will see that in the force profile too. And this is just showing low levels of expression, providing it's uniform, are sufficient to improve muscle function. There's the wild type MDX. Freddy, Fergie, and Fiona are just different transgenics going from low expressors to high expressors, and all give substantial amount of rescue of the muscle function. So how can we increase the levels of utrophin? And so we set up a transcriptional screen where we took one of the promoters, and exactly like dystrophin, but in different places, utrophin has several promoters, and we focused on this promoter A, so-called here, which is predominantly expressed in muscle. Promoter B is predominantly expressed in endothelial cells. And we have two non-coding exons at the 5' end of the gene. You can see the start code on here, ATG. And so we put in a luciferase gene so that we could read out directly from this gene. And this was upstream from the regulatory elements that are at the 5' end of this gene. And the N box is the element which is responsible for the expression of utrophin at the neuromuscular junction. And that was first characterized in the acetylcholine receptor genes, which are also localized at the neuromuscular junction. If you mutate the NMJ, then you no longer, sorry, if you mutate the N box, you no longer get the MNJ localization. So what we think is happening in early fetal life is that there are transcriptional factors which override this signal here through some signaling pathway, which allow it to be expressed at the sarcolemma as well as the NMJ. We also know that if you damage muscle, the fetal program is recapitulated, and again, this N box regulation is overridden, and you get spreading of utrophin at the sarcolemma. So we did a screening of several 100,000 of these molecules, and we identified a drug in collaboration with Summit Therapeutics called azutramid, and found that azutramid increases utrophin levels by at least twofold in these cell lines. We then went into the MDX mice, and this is EDL and TA shown here. And you can see MDX plus azutramid gives a substantial increase with uniform staining around the sarcolemma. Does it actually spread along the whole membrane? And you can see this by doing not only longitudinal sections, but transverse sections. And you can see here, is along the whole length, we've got positive staining of utrophin. So utrophin overexpression at this level appears to stabilize the membrane and reduces the rate of degeneration. This is normalized force increment, the control, exercised MDX, and exercised MDX plus azutramid, again showing the prevention of the disease. So I'm not going to review all the data, it's now been published. There's no question that in the MDX mouse, at this stage at least, we were able to modify the disease profile. And therefore, this was something that we could think about taking into clinical trials. What we needed was a marker to show that we really had prevented the muscle damage. And for that, we developed developmental myosin as a marker of regeneration. The MYH3, the developmental myosin, is expressed in MDX muscle. It's re-expressed in regenerating fibers. And therefore, treatment with utrophin, we ought to be able to see this developmental myosin going down as utrophin goes up. That just shows you the profile. I'll miss that out in the interest of time. This is the wild type. This is DMD dystrophin, developmental myosin, and the merged. So there's no developmental myosin, but you can see muscle damage is clearly shown in the regenerating muscle. You can be seen by looking at this marker in the MDX. If we then take a exon skipping, a PIP PMO, which we collaborated with Matthew Wood's lab in the department on, you get exon skipping, so you get production of dystrophin again, you get downregulation of that developmental myosin, as you can see. So this is a very good marker in the MDX mouse, and we can also increase utrophin in our transgenics, and I'm just going to show you this, the MDX Fiona here. We compared to the MDX wild type, again, we get downregulation of this very important marker. If you take away dystrophin and utrophin, incidentally, you get a very bad, severe muscle disorder, and that, again, is exemplified by the high expression of this particular marker. This is consistent with what you see in patients. This is a study that Summit Therapeutics undertook in collaboration with Flagship, where you can see there's a trend between the amount of developmental myosin that's expressed versus the mild defected patients. So a BMD patient is at this end, and you get predominantly DMD patients at that end. It turns out that these are very severely affected Becker muscular dystrophy patients in this context. So Flagship developed an automated way of measuring the developmental myosin fibers and the utrophin expression. Laminin alpha 2 was used as the control marker. And we carried out a phase 2 clinical trial, or rather Summit Therapeutics took this to a phase 2 clinical trial. And we were very excited because after 24 weeks, that was the baseline muscle biopsy. And after week 24, you can see that we have indeed reduced developmental myosin expression, and that's shown in this graph here. Unfortunately, after a full 48 weeks, this particular data was not reproduced, and we could see no difference between the controls and the treated patients. So we knew that azithromycin exposure decreases with repeated dosing in healthy volunteer and markedly in DMD patients. It's rapidly turned over. As you can see, it undergoes significant metabolism, and therefore repeated exposure leads to a significant reduction. So the question is, can the lack of sustainability of this clinical response, which we were so excited about at 24 weeks, could it be that it's a turnover of the drug and it's bad metabolism that's causing the problem? Can we establish that azithromycin engages with the target, which really does upregulate utrophin? And can we therefore use this in the future, perhaps by developing a drug which doesn't have that bad metabolism as we've seen? So I'd just like to now present data that was generated by Isabel Wilkinson when she was working in Angela Russell's group, and all this work has been carried out in collaboration with Angela Russell and Steve Davis in chemistry. So we used photo affinity labeled or biotinylated probes, and we used the usual ChIP-seq, RNA-seq, proteomics, and Discoverix. And I'd just like to go through these data with you. The first was an RNA-seq analysis, and this first of all showed that the aryl hydrocarbon receptor, AHR as I'll call it, was upregulated and its repressor was downregulated after ezetimibe treatment. And you can see that peak here. So Isabel went on to make an affinity probe. This is ezetimibe. This was the affinity probe that she used, and this is the negative control with a different side group here. Now I put the reference in there because we've just recently published these data. Probe validation, we just made sure that we didn't affect anything in the development of the muscle. And then we went on to do the target ID biochemical chemoproteomics. And you can see here that the AHR is coming up as a positive in this particular assay. I don't have time to go through that in detail. What does the aryl hydrocarbon receptor do? Well, it's been involved in a whole host of different diseases: cancer, immune diseases, xenobiotic responses. Is it sensible that this particular receptor could be connected to the expression of utrophin? Well, yes, because the agonist PCB126 was found to decrease utrophin in a completely independent study in the literature on rat liver progenitor cells. Resveratrol upregulates utrophin and decreases inflammation in the dystrophic mouse, is also an AHR antagonist. AHR signaling regulates PGC1 alpha, a utrophin transcription factor that was extensively looked into by Bernie Jasmine's group, and also it regulates the COX-2, and COX-2 inhibitors also increase utrophin expression, again work from Bernie Jasmine's group. So looking again in more detail at this experiment, this is the probe, positive control, this is the negative control, and you can see that we can compete out, yes, it's ezetimibe, we can compete out this using a competitor. And we can also show that ezetimibe binds to a purified receptor complex in vitro. This is shown here with the binding curve. But perhaps even more dramatically, we can see that ezetimibe blocks the nuclear translocation of the receptor in muscle cells and therefore negatively regulates the receptor genes. Now that's DMSO, this is ezetimibe, and you can see here with the receptor, we've only got it in the cytoplasm. This is an agonist where you can see it's the receptor is in the nucleus, and if we have the mixture of the two, if we had ezetimibe and the agonist, we then get again blocking of the receptor in the nucleus. If we look at the downstream genes of the acetylcholine receptor, we can see that these are downregulated in response to the treatment with ezetimibe, which is entirely consistent with ezetimibe blocking this nuclear translocation. Now, if that's the case, of course, we would expect other, and there's quite a few of them available, structurally distinct AHR antagonists to give a concentration-dependent increase in utrophin. They don't all do. This is one that doesn't, but you can see that indeed we do observe this with other AHR antagonists, and this just shows you the protein. Importantly, these experiments were carried out in a human cell line, not a mouse cell line. So these data extrapolate to the human condition, which again is consistent with us being able to use this pathway in human as well as mouse cells, and obviously in clinical trials as well as the MDX. So in the mechanism of action study, we've now demonstrated that these studies converge on AHR as a target of ezetimibe. We know what the binding characteristics are, and therefore ezetimibe behaves functionally as a AHR antagonist. So now we're continuing our screens, going back to our transcriptional screens to see if we can find other molecules which will increase utrophin, and also looking at other antagonists of AHR to see if we can find other relatives. And our new screen does indeed identify new molecules which didn't come up in the initial screen. That's the initial screen with just this portion of the genomic locus. If we actually knock in the luciferase reporter gene and have it in its whole genomic context, then we begin to identify novel compounds. So at the moment, we're using that genomic screen to identify molecules that may have a different mechanism of action from azutramid, which means we can use two drugs together. And we're looking at other small molecules which target the pathway involving AHR. Finally, does the combination, none of these therapies, whether it's exon skipping, gene therapy with a mini gene, or utrophin replacement, will completely rescue the phenotype. If we try and combine these therapies, do we get a better result? Or is it that we are saturating the membrane and that we can't add more utrophin, for example? So what Simon Guyu did in my group was to take transgenic mice and PMO, that's an exon skipping oligonucleotide treated mice, to see whether we could do this additively. And I'm just going to go to the last slide here because I want to give time for questions. This is the MDX. This is our transgenic that expressed a small amount of dystrophin. And this is PMO treated MDX Fergie. And you can see that together they act synergistically. If we do the same for the high expressor utrophin, again, this is PMO treated MDX Fiona. Again, we get a greater rescue. If we look at the level of dystrophin in a wild type mouse highly expressing utrophin, we do downregulate dystrophin. So they do compete at very high levels, but at the sort of levels we're looking at in exon skipping treatment or gene therapy treatment, then it's clear that we will be able to use combination therapy. And that again, and that the dystrophin and utrophin can co-localize at the membrane. This is PMO treated MDX, there's dystrophin, and that's merged. This is MDX Fiona, high expressing utrophin, and then exon skipper oligonucleotide treated MDX Fiona. And you can see the merge shows the co-localization of dystrophin with utrophin at the membrane. So the combination therapy has greater therapeutic potential. So I'll skip that. Coming back to genetic approaches for DMD therapy in 2020, I think we will see improvements of exon skipping. Gene therapy, as I've explained to you, is now looking very exciting in clinical trials. And if we can find a modulator with better metabolism than azutramid, then modulation of utrophin with a small drug can play a major role in the therapy of DMD. And not only that, this will be easily applicable for all patients around the world. And it's clear also that because each approach does not completely restore function, combination therapy should demonstrate significant clinical improvement. None of this work could be done without collaborators. And one of the things that I've learned in doing translational research that is not just the academic labs, but also biotech, and we've had help from Sarepta, but also how important it is to link in with the charities and the patients to think about what their needs are at the very beginning of your project, so you can develop good candidates for the disease. If I put down all the members of my group, they would take several pages, but these are the people who have made significant contributions, particularly in my group: Simon, Sarah, Ben, Aaron. Sarah and Ben and Aaron are still working with me, Kelly, Adam, Lee, and Hannah, and all the people in Summit, and the chemists without whom we couldn't have done any of this drug development: Steve, Angie, Graham, Maria, and Isabel. There's the picture of the chemists outside the my building. And finally, at the point I've just made, we couldn't do it without the inspiration and the generosity of the charities that support research into this disease. So I'd like to finish there with a big thank you to them. And again, I'm honoured to give this lecture to the Biochemical Society. This is a very exciting field. And I think for the first time in my life, I can see the light at the end of the tunnel. Therapy is an effective therapy is nearly there for Duchenne muscular dystrophy patients. Thank you.
Thanks a lot, Kay, for this amazing talk. I think it really exemplifies how therapy discovery integrates so many different disciplines, in your case, genetics, biochemistry, pharmacology, medicinal chemistry, and also emphasizes the teamwork behind those discoveries. So we do have a few questions which I'm going to run through. So I'll just start with the one, and I apologize in advance if it's been already covered in the talk. Since those were sent during the talk. So the first one is, in this case, the re-expressed dystrophin is immunogenic. How do you propose to deal with this problem, please? And I think that probably is the reason why you focused on Utrophin. So I just wondered if you could answer this.
Yes, I think that one of the problems with AAV dystrophin is that if you re-express dystrophin, it's internal to the muscle cell, so we're not sure how big a problem that will be long-term in muscular dystrophy patients. But there's no question that they do mount some immune response. I think the difficulty is if the virus is gradually lost and then you re-administer a virus, then you'll have an even greater response to the virus. So you'll have to either use a different virus or a different serotype. So that's the challenge of AAV as it stands at the moment. It may be that you could get immunosuppressant strategies that would circumvent that. But that's one of the drivers that Hansel Stedman's group used when they did a Utrophin mini-gene, because if you deliver a Utrophin mini-gene, you don't have an immune response except to the virus. So at least you get rid of part of that effect. But it's a very important problem, particularly at such high doses.
And related to that, I believe in Oxford, they use the ChAdOx, the chimpanzee adenovirus vector for the COVID-19 because you wouldn't have any pre-existing immune response. Is this something that's been considered?
Well, yes, and I think one of the reasons that Sarepta have been so successful in their gene therapy trial and Solid have had therapeutics biosciences have had side effects is because they used AAV9 and Sarepta used a different type of construct, still AAV, but a different serotype, which turns out to be less of a problem because Pfizer also in their trials have used AAV and have seen adverse effects. So the choice of the virus is going to be key, and maybe engineering capsid proteins in the future that many biotech companies are working on will help with that also.
So the second question is really interesting. I look forward to the answer to this one. Why does it affect cognitive abilities?
Everyone asks that question every time, and there is no straight answer to that. We do not know what dystrophin does in the brain. So you could say, well, maybe there are certain mutations that predispose. So if you use the carboxy-terminal mutations, for example, point mutations, maybe you get more severe cognitive impairment. The answer is we don't. There's a general shift towards mental impairment, and it's only in about 30% of patients. So we don't. It's very poorly understood, if understood at all, actually. So all I can say is, because this is biased because I'm looking at utrophin modulation, if you could come out with something that got to the brain as well, cross the blood-brain barrier, then if you get in early enough, then maybe you could do something about that as well.
I forgot to name the people who forward those questions. So the first one was Darius Goreski, and the second one is Heather Churchill. So on to the third question by Mazar Mustaq. Many thanks for these valuable information. Just curious, what happens to calcium channels in DMD?
Well, yes, they are altered, and in fact, what we've shown is, and I haven't had time to do that data, is that transgenes do replace the function of those calcium channels, but they haven't been very well studied in the sort of therapy models that I've been describing today. Whether you get full correction. So that's something that I think that we would need to look at more carefully as we looked at the efficacy. Why aren't we getting full rescue? That would be a very important question to look at the function of some of these calcium channels, because it may be that we're not actually reversing some of the loss of function that you see in the patients.
So the next question from Joshua Lawrence Bautista. What are the biochemical criteria for you to classify if it is a DMD biomarker or not?
The biochemical.
I suspect you have potential clinical biomarkers for your studies. I guess that's.
Well, again, there are not very good clinical biomarkers for DMD. That's been one of the problems. And in fact, for the clinical trials, the endpoint had been the level of dystrophin, which isn't a very good biomarker. And people have done all sorts of experiments, and we've published experiments. Matthew Wood Lab have on somologics, proteomics profiling of DMD patients before and after treatment. And there are one or two markers, but they're not 100% consistent. So there is no reliable biomarker that we can use in the clinic. If there were, we'd all be using it, and we wouldn't be using a 6-minute walk test, which is what we started with, or even the North Star score, which is what Francesco Montoni and his colleagues pioneered, which is how fast you walk upstairs, etc. If we had a biomarker, it could transform progress, but we do not have one at the moment. Or not a reliable one anyway.
There are loads of questions. I'm not sure we can go through all of them, but so the next one is Shamshad Cockcroft. Are there any known disease modifiers that lead to milder forms of disease?
Yes, there are other pathways that Luke Uncle's looked at. There are dogs which actually have the dystrophin, don't have dystrophin, but are very relatively mildly affected. So there are several pathways now that people are looking at which might ameliorate the disease, and that those are pathways that can be targeted. And in fact, those sort of developments are in hand by other groups around the world. I mean, modifiers is certainly one way to go.
Thank you very much. Next question is again from Darius Gorecki. And the question is, was the AHR study performed in myoblasts or myotubes, please?
We did it in myoblasts.
And if I may, I had a question on AHR. What, if you antagonize AHR, do you expect any secondary effects? Because I believe it has a role in regulatory T cells, and you want to agonize it.
Lots of effects. So I think that's the challenge of getting another antagonist. We didn't see any secondary effects, but obviously after 24 weeks we might not have done. But that is the caution that you would have to say. What I think we can say is we know what the pathway is now. We know that azutramid did work because we know what the target is. Is there another part of that pathway that we can target, which would increase utrophin without any of the side effects that receptor might have?
Okay. Next question from Lucia Moro. What do you think about gene therapy with CRISPR? Can it be safe enough?
I think eventually all CRISPR will be safer because the technology is advancing so fast. It really must be promising. I think the difficulty there will be how much better will that be than the AAV microgene that Sarepta and Pfizer and Solid have got out there now. And the reason I say that is because CRISPR will actually, you still have to have a mini gene, you still have to have AAV delivering the CRISPR and slicing out the gene. So in certain circumstances, in correcting a single base mutation or very small deletions, CRISPR may be better. But whether someone would pay for all that development for a very small proportion of patients, I'm not sure. And that depends on the development of the technology and how rapidly that will happen. But if you're already getting a very good result with an AAV microgene, would you use 2 AAVs to get all the CRISPR machinery in to do it? At this stage, you probably wouldn't. So we'll have to see how the field evolves in the CRISPR technology space.
Makes sense. Next question from Rumiko Ochai. I apologize if I don't pronounce these names very well. Can ezetimibe ameliorate not only skeletal muscle function, but also cardiac function?
Okay, we've done some very preliminary studies on this one, and the answer is yes. But we've done those experiments in the MDX mouse, which really doesn't show very strong cardiac phenotype. So I wouldn't like to say that we've done any studies that would reflect what would happen in man. There's a hint that it would, and so that suggests that increasing utrophin at those levels will help. But I think whether that particular drug would do it effectively in the heart, we have no idea what levels we would need. Actually, we don't even know much about the dystrophin either. But again, these sort of things will come out of the clinical studies using utrophin microgene therapy and the dystrophin microgene therapies.
Okay, thanks. Next question from Lisa Chakrabarti.
Chakrabarti, I know that one.
Oh, okay. At which point in the course of disease would you expect to start therapy? Is sooner better? Do you envisage testing all baby boys?
Yeah, I missed that part out of my lecture. There's a balance. I think the moment there is an effective therapy, we'll have to screen all births so that these tragedies of multiple boys in families being affected is suddenly realized too late. So it's already been piloted in the States, in Holland, actually in this country too. If we could screen this, know that the mutations are there, the earlier you start, the better it is. And that's true in the MDX mouse and the dogs also, as early as possible.
Okay. Another question from Rumiko Ochai. In DMD patients, you mentioned that utrophin is already upregulated. Do you still expect twofold increase can be effective in those patients?
Yeah, that's a very good question, because a lot of the DMD patients do show increases of utrophin. The trouble is what you see is the rapid turnover of utrophin, because you're seeing utrophin's upregulated in those regenerating fibers, and then it is rapidly turned off. The difference with a drug is that we're producing it at enough levels so that it's stable at the membrane, so it isn't turned over. So there's a difference. There isn't a difference in level, but there's a difference in the stable level of the utrophin protein on any muscle fiber, and that's what makes the difference.
Question from Mario Pavez-Gianni. What is your option about clinical impact over older patients? So patients, over 20 years old that present a higher muscle damage. So can you have a clinical impact on older patients is a question.
I think that, yeah, it's a very good question, and I think it's going to be very difficult. I think you can perhaps arrest the disease, but then you certainly, unless you're well, gene therapy might do it. If you could infect these stem cells and regenerate more muscle, that would be possible, but that's very challenging indeed. So I think the most you could hope for would be that you might slow the progression in the later stages of the disease. But certainly, again, I think once some of these are approved, we need to get into those non-ambulatory patients and older patients in their 19, 20, 21, early 20s to see whether we could do exactly that.
Okay, there are some more questions. Unfortunately, I don't think we can cover them all. We're getting to 3 o'clock. So that leads me to thank you again, Kay, for this wonderful talk. Also, thanks to all the people that have attended and all these really nice questions. And I apologize I wasn't able to go through all of them. So just like to also remind you that we are interested in suggestions for future topics and speakers. If you have any idea for a webinar, you're invited to submit a proposal, a short proposal for an idea, and you can find those proposals on the biochemistry.org/webinars website. So the next webinar is on the 6th of August at 3 p.m. and it will be a panel discussion on non-academic careers for molecular bioscientists. And you can find more details of those on the website. So again, thank you very much everyone for attending, and thanks again, Kay. That was really great to be able to chair this wonderful lecture. Thanks and bye.
Bye-bye. Thank you.